Hello, my name is Marie-Charlotte Schönerich and I'm a postdoc in Jill Banfield's lab at the University of California, Berkeley. And I have the pleasure of presenting to you our work on the conformation of Borg extrachromosomal genomes, analysis of their methylation patterns, metabolism, and tandem repeats. Methane emissions are driving climate change, and we're very aware that microorganisms are key in the cycling of these greenhouse gases. So methane originates from many natural sites, such as natural wetlands, agricultural sites, waste sites, but also from fossil fuels and natural emissions from the oceans and gastrointestinal tracts of insects, such as termites, for example. So many of us in the lab study these sites. For example, a natural wetland just 2 hours north of Berkeley is a site that we studied for a long time because we're very interested in finding out who are the microbial players that are important in the cycling of methane. So this is a very simplified depiction of the cycling of CO2 and methane, and we're predominantly interested in the anoxic processes, so these that happen down here. So we know methanogenic archaea produce methane, and the key enzyme for this is the methyl-CoAM reductase, or MCR complex. There are also archaea that have evolved from these methanogens to reverse the process, so to oxidize methane back to CO2, and they basically use the same enzymatic machinery. And these are the organisms that we're particularly interested in. So using genome-resolved metagenomics, we were able to discover completely new DNA entities that seem to associate with these methane-consuming archaea. So the host of these novel DNA elements that we term Borgs, they oxidize methane to CO2, and electrons are then transferred onto extracellular compounds. And besides having a main chromosome, many of these methane-consuming archaea called Methanoperedens seem to have Borg DNA within them. And what's very exciting about the Borg DNA is that they carry important genes from these methane-consuming archaea. So some Borgs, for example, encode the methyl-CoAM reductase that is important in oxidizing methane to CO2. And the first discovery was published by us in Nature just last year. And at that point, we had 4 Borg genomes. And we saw that, or 4 complete Borg genomes. We knew of the existence of more, but it was hard to complete these genomes. So at that point, we knew they're always linear, they're composed of double-stranded DNA, and they're up to 1.1 megabase parent size, so huge. And they're clearly not part of a microbial genome. But what are they? We could certainly see that the Borgs have a very conserved genome topology, so they're linear, they're terminated by long inverted repeats, and they're always composed of 2 replicors, and each replicor carries all the genes just on one strand. Another striking feature that they have is that they are scattered with these pervasive nucleotide tandem repeats. So this nucleotide tandem repeat is a piece of DNA that's repeated in a head-to-tail pattern, and every instance of such a red line that indicates such a repeat is a unique nucleotide sequence. So this means that they evolved recently. Moreover, we see in mapped reads files that these regions are rapidly evolving, just like CRISPR loci. Of course, begging the question, what is the function of these rapidly evolving nucleotide tandem repeat regions? In order to answer that, we did more in silico analysis and found that about half of them are within open reading frames, and the other half are found in intergenic regions. And when they are in open reading frames, the tandem repeats introduce amino acid tandem repeats, and these are often intrinsically disordered regions. What is intrinsic disorder? It's a region in a protein that lacks a 3D structure. So it looks a bit like this loopy extension here in this GIF image. So it can take on a 3D structure when it is bound to its interaction partner or post-translationally modified. And we hypothesize that the role of these tandem repeats is in gene regulation, but also protein localization and protein function. Now, in order to illuminate more about these Borgs and discover more Borg genomes, we decided to team up with Oxford Nanopore Technologies, specifically the US team. And we were able to find that nanopore reads assemblies actually confirm the overall Borg genome topology of Illumina-derived curated complete genomes. So you can see an alignment, a nucleotide alignment of the nanopore assembly to the Illumina assembly, and it overall confirms the topology, but it does flag some really consistent discrepancies. So these are indicated by these red lines here. And we found that these discrepancies are almost always found in the nucleotide tandem repeat regions. So these are flagged down here by these colorful little triangles. So we found that in these regions, the sequences were different. And I've shown some examples here. For example, this repeat region, in the bottom, you see the Illumina assembly. It is missing a piece that we find in the nanopore assembly. So in this case, we find that there's one additional repeat unit that is actually within an open reading frame that encodes a polymorphic outer membrane protein. A similar scenario is here in this case study too. Here we actually have a longer stretch of several repeat units that were missing from the Illumina-based assembly. And then there is even a more extreme case where we really have about 20 tandem repeat units that were missing in the Illumina-based assembly. And this makes sense because we are aware that just using short reads, during the assembly, it often happens that these nucleotide tandem repeat regions are collapsed. So this is a great example that shows why it's important to also use long reads, especially if you want to know the actual overall genome length and you want to resolve these nucleotide tandem repeat regions. So besides confirming the existence and overall topology of 5 existing Borg genomes, we were also able to discover 7 new Borg genomes in the nanopore data. And this was actually led by Lin Li, a bioinformatician here in the Alameda office. So what she did was she mapped contigs in terms of their GC content. So Borgs always have a very characteristic GC content, and also mapped the coding density and the coding switching. So all the Borg genes are always encoded on one strand. So by plotting these features, she was able to identify all the contigs that match existing Borg genomes. So these are highlighted in green circles, but she also found in purple circles these new contigs that correspond to new Borg genomes. So now we have a list of 17 close to complete genomes that are shown here. So the new genomes that we got through nanopore sequencing are those that correspond to cobalt, Borg, amethyst, amber, ruby, iris, viridian, and emerald. And this is really fantastic because it allows us to now establish the core Borg proteome. So we have these 17 Borg genomes that are close to complete or complete in most cases. And we can look at what is the inventory of genes that is present in all of them. And another great accomplishment was that we were able to curate the first complete Methanoperedens Borg host genome. And now that we have the closed genome, we see why it's so important, or why these genomes are often so fragmented. That's because they are scattered with hundreds of transposases, making assemblies really, really difficult. But now that we have this complete Borg genome and Borg host genome, we can work on the metabolic reconstruction and really look at what is the gene inventory of the Borg that is complementing the metabolic gene inventory of their host. And finally, we were also able to illuminate the distinct and pervasive modification patterns in Borg and host DNA. So what happens during nanopore sequencing is that nucleic acids are being translocated through these protein pores, and what is detected is an electrical current. So this electrical current will be distorted if the nucleic acids that are translocated through the pore are modified. So if they're methylated, for example. So if you then sequence amplified DNA that of course lacks this modification and overlay this signal to the sequencing of native DNA, you will get differences in the current that are actually then visualized as these squiggles, and they indicate that in this case, this region corresponds to a region in the genome that is modified, probably methylated. And using this approach, we were able to find that Methanoperedens have 6 distinct methylation motifs, and really surprisingly, the Borg genomes consistently have 2 methylation motifs that are just composed of 2 nucleotides, so TC and CC motif. And that was very surprising to us because usually you have longer motifs, and this is a little bit reminiscent of eukaryotic CpG islands. Now, the purple Borg also has a 3rd methylation motif composed of GAA, and we believe that the CC and TC modifications could be 4MC modifications, whereas the GAA modification is likely a 6MA modification. And by having just these 2 nucleotides corresponding to a methylated motif, this means you have hundreds of these across the genome. And in fact, even more than every 10th nucleotide is composed or is within such a motif. So potentially we have really a hypermethylation of this DNA. So that's very exciting to us. And another analogy also to the CpG islands is that 71% of the motifs are found in non-coding regions. So from this, we hypothesize that these very peculiar modifications in the Borg genomes could be important in regulating gene expression. And with this, I'm at the end of my talk, and I would like to really thank all the amazing collaborators from Oxford Nanopore Technology here in the US, and of course, Jill Banfield, our group lead, all the amazing members from the Banfield Lab, our funding buddies, and you for your attention, and I'm excited to take questions. Thank you.